All right, I think we can get started. So welcome everybody. Thank you for joining us today. I'm very excited to be here. My name is Ryan McDonald. I am the marketing director here at Elixirgen Scientific. I will be hosting this session, and I'm joined by two really great speakers, which I'm really excited to speak with and hear from today. For our session titled Unlocking the Secrets of Alzheimer's, Epilepsy, and More with Cutting-Edge iPSC Technology. I'm joined by Dr. Cara Gordon from Vala Sciences, and I'm joined by Dr. Masashi Tabuchi from Case Western Reserve University. Two really great researchers doing two really cool things with our neurons, and I'm really excited to hear from them today. So for those of you who haven't been to our webinars before, this is kind of how it goes. We'll have our speakers, and throughout the webinar, if you have any questions, you can just put those within the comments at any point. Please, we ask that you include the speaker and the slide number just so that we can pull up that slide or get the right speaker to answer your question. And you can also email your question if you're not comfortable putting your question for whatever reason within the comments. You can always email the question to us at questions@elixirgensci.com, and we'll get those answered for you. And if your question is not addressed during the webinar today, we usually get to all of them, but if it's not, we'll follow up with you in an email and make sure that we get those questions answered for you, or kind of work with one of our speakers here to get you the information that you guys are asking for. So without further ado, our first guest speaker is Dr. Masashi Tabuchi from Case Western Reserve University. He got his PhD in engineering from the University of Tokyo, and then did his postdoctoral fellowship at Johns Hopkins University right here in Baltimore. And he's currently a faculty member in the Department of Neuroscience at Case Western Reserve University School of Medicine, leading a laboratory focused on understanding the biophysical basis of introspective neural processing and neurological disease. And the goal of the Tabuchi lab is to gain fundamental insights into how the nervous system generates computational mechanisms to regulate our internal states, with the long-term goal of identifying therapeutic targets for neurological diseases. And without further ado, our first guest speaker, Dr. Masashi Tabuchi.
Okay, thanks so much for introducing me, Ryan. So I'm very excited to be able to speak here today. So as he said, my lab is studying neural coding mechanisms of sleep, primarily using Drosophila melanogaster fruit fly. However, thanks to the amazing product of Elixirgen Scientific, my lab could also start using human iPS cell deriving neuron. So today, by combining Drosophila and the human iPS cell, I'm very glad to share our first result of a human iPS cell obtained from real Alzheimer's disease patient, which their experiment design are actually based on discovery from Drosophila. So sleep disturbance is an extremely common symptom in Alzheimer's disease. So as you can see this graph, the reduction in slow wave sleep and increase of brief awakening during nighttime are commonly observed in Alzheimer's disease patients, with strong correlation to the degree of their cognitive impairment. So we want to know how these types of disturbed sleep regulation can be induced by Alzheimer's disease pathogenesis. So to do that, first, as I said, I have to introduce about our Drosophila model before talking about iPS cell. So regardless, Drosophila or human circadian clock neuronal activity is thought to be one of the most important components of sleep regulation. And as you can see this slide here, the circadian rhythm can be generated by negative feedback loop of molecular clock, and this oscillation provides circadian regulation of sleep-wake cycling, and which is actually a cause of electrical activity pattern of these neurons. And in Drosophila, there is a specific neural cell types of circadian clock neuron called DN1. And this DN1 neuron is projecting to downstream arousal circuit, which is called PI neuron. And these synapses are the most important neuronal pair controlling circadian regulation of sleep in Drosophila. And as illustrated here in the graphics, we can record their electric activity in vivo, behaving fly, so that we can associate how this electrical activity is causally related to actual sleep, behavioral sleep. So I want to talk about neural dynamics, which is actually related to aging. So we previously found that activity pattern of this DN1 clock neuron is especially important for regulating sleep quality. And this activity pattern dependent sleep quality regulation is actually also age related. So in young animal, which is shown left side, the DN1 activity become very regular during nighttime, which is shown in blue. On the other hand, during daytime, the activity is very irregular, which actually promote arousal. So basically, this circadian difference of activity pattern between daytime and nighttime is very important for sleep quality. But as you can see here, if we just repeat same recording in aged animal, actually such circadian difference between daytime and nighttime are no longer observed. So as you can see, both daytime, nighttime activity pattern are actually just like very irregular, which is actually based on very unstable dynamics of neural membrane potential. So basically, as you can based on this observation, we propose that neuronal stability is really important to achieve very precise sleep regulation. Then we wonder, or maybe AD pathogenesis can also make neuron unstable, just like what we observed in aged neuron. So to test this hypothesis, to test this if pathological tau can also bring similar sleep instability, based on that the fact that Alzheimer's disease is known to be age associated. In Drosophila, we utilize specific form of neurological pro-aggregant tau having mutation in the second 4-repeat domain. So we found actually in Drosophila expressing this pathological tau in DN1 neuron showed reduced sleep quality as well as quantity. So basically, this result really suggests that our hypothesis is really correct. So basically, potentially unstable neural dynamics may induce sleep instability, which is caused neurodegenerative disease. But so we want to show how pathological tau cause dynamical instability of this DN1 neuron. So to quantify dynamical instability or stability of biophysics of neuron, I want to bring the new model based on probabilistic state transition model. So as example, let's talk about kind of weather forecast. So as you can see, now I just aligned stable weather or unstable weather, and these kind of weather state can be aligned as a time series, and we can segment. We can perform segmentation based on discrete moment to moment. So that means time structure can be sunny and cloudy, and sunny, or something like that. So basically, then if we align these things as discrete Markov model, just like shown here, we can visualize how weather can be stable or how weather can be unstable. So in this example, as you can see, stable weather can show very strong persistence on sunny state, right, which is shown in left side. On the other hand, unstable weather, which is bottom line, actually there's no preferred state between sunny, cloudy, or rainy, instead, actually, their state change is very rapid. Sometimes it becomes sunny, and the next moment it becomes rainy, and next moment it becomes cloudy or something like this. So we think these types of modeling approach can be generalized and can be applicable in neural nervous system. So as you can see here, now on the left panel, I put spike train as the activity pattern of neuron. So as you may know, neuron fire action potential, and this spike train can also be classified based on several parameter, like rapidness of action potential onset or stability of action potential offset rapidness. But by using any types of biophysical parameter, we can classify, just like in sunny or rainy state. We can define state of individual action potential. In this example, as you can see, I could classify group A in first spike train, and the next spike train also group A, and the next spike train also group A, but next spike train now I classified as class B, which is different from class A. And something like this, we can also further classify what types of action potential groups are there, and how many cluster we want to generate is actually up to our criteria. But in this example, we could classify 4 different states, right? Group A, B, C, D, and as you can see, as the left side is a stable example. So then it tend to have more group A, and group A tend to have strong persistence of state. So they have very strong recurrent connection recurrent dynamics that A try to stay A again even in next moment. So this is an example of how stable state can be represented in nervous system. On the other hand, the right side, it's more unstable and rapid state transition from D, B, C, B, A, C, D, B. These things happen. So this is defined as very unstable neural dynamics. So based on this discrete Markov model transition modeling approach, we decided to test if this neural state can be altered by pathological tau. Yeah, the answer was actually yes. So basically, when we express this pathological tau in Drosophila circadian clock neuron, yes, it actually showed the neuron become very unstable. So first, as a biophysical parameter, we quantified the Lyapunov exponent based on membrane potential dynamics, and we found a significantly increased Lyapunov exponent are there in the presence of tau in DN1. And again, we implemented a discrete Markov chain model to compare, how state transition can be there between control and tau. And as you can see, in control, we found a very strong state persistence in group 2, which is never observed in tau expressing DN1 cell. As you can see here, tau expressing DN1 cell shows very rapid state transition between these 3 groups, which is also quantified by the increased local variation of action potential statistics, which is also shown as CV2. So based on this, now I can say, expression of tau makes neuron very unstable. But then we decided to test if anti-epileptic drug actually can rescue these types of dynamical instability in DN1 clock neuron expressing tau. Yes, actually it works. So basically, as you can see, if we apply anti-epileptic drug such as levetiracetam or brivaracetam, actually we found the Lyapunov exponent are significantly decreased compared to DN1 expressing tau alone. And then, as you can see in hidden in Markov discrete Markov chain model, compared to tau alone, if we apply levetiracetam and brivaracetam, yeah, it's not perfect, but at least we could found some rescued state persistence in group 2, which was observed in control. As you can see, in the presence of levetiracetam, brivaracetam, state 2 shows some kind of recurrent network, a recurrent connection that they try to stay in same state even in next moment. So, and similarly, we also found the increased local variability of action potential observed in tau was significantly decreased in tau in the presence of levetiracetam and brivaracetam. So basically, these results really suggesting that anti-epileptic drug is actually useful to reduce the unstable state of circadian clock neuron dynamics, which is induced by a pathological form of tau. And we wanted to know what is the biophysical origin of these dynamics, and at least one of the target can be voltage-gated sodium channel. So we perform voltage clamp experiment to quantify inactivation status of voltage-gated sodium channel in DN1, and we first found inactivation status of voltage-gated sodium channel also become very unstable in presence of tau, which is actually rescued by brivaracetam. So I think at least because sodium channel is definitely one of the major components to make neural dynamics. So that's why probably we think this is one of the biophysical origin creating this dynamical instability. And yes, and this is a noise analysis of this sodium channel. So basically, this noise analysis is telling that significant noise, instantaneous noise peak was found in tau, but that is suppressed, at least reduced, significantly reduced in the presence of brivaracetam. So I think so. So basically, decreased noise, reduce instability of neural dynamics in DN1 cell, DN1 clock cell. So then, so now these are Drosophila part. Sorry for making a lot. But then now I want to move, human iPS part. So, thanks to Elixirgen Scientific, we could culture 5 individual of iPS derived product. So basically, 3 control and two control and 3 AD patient were used in the dish to record the action potential dynamics. And similar to Drosophila experiment, we also found, we applied the same discrete Markov chain model and found actually AD patient also has unstable rapid to state transition compared to control. So control has a state persistence in specific state, but that's not observed in ADE. And we found that levetiracetam or brivaracetam application in the culture media is actually very powerful to try to suppress this rapid to state transition. We also perform other types of computational model, which is based on Gaussian mixture model. So in this case, we focusing on adjacent interspike interval correlation to analyze what types of secondary structure is there. So basically, what we found here is, in control, there are basically 2 major preferred state are characterized in the adjacent interspike interval profile. But as you can see here, on the other hand, maybe AD model, AD iPS cell tend to have more unitary space in this GMM map. So I think maybe some second we can say that this second order structure is actually lost in AD. And then we found that this second order or first order difference between control and AD also rescued by levetiracetam and pluripotent. As you can see here, in the presence of levetiracetam and pluripotent, even in AD patient iPS cell, actually the two groups of GMM profile can be observed. So I think this really suggests that levetiracetam, pluripotent achieve these types of recovery by making neuron more stable. And yeah, this is more basic characteristics of activity pattern. But actually, as you can see, we never found a broader global change in terms of mean firing rate. So as you can see, as in terms of neural activity amount, actually we never find any significant difference between control and AD. So we only found local irregularity of interspike interval. So basically, I think this is some kind of important concept that microscale dynamics is actually containing very important meaningful information kind of telling how pathology is generated by AD pathologies, which is actually never found in more major phenotypes such as mean firing rate of action potential. So basically, to summarize this, we first by using Drosophila, we found that tau makes sleep unstable by making circadian clock neuron more unstable, which is actually based on the increased sodium channel noise. And these altered parameter and increased instability is actually also observed in AD patient-derived iPS neuron. And finally, we found these phenotypes are actually can be suppressed by administering anti-epileptic drug. And then I think Dr. Gordon will talk more about epilepsy, but my lab is also interested in epilepsy because, as you can see here, epilepsy and Alzheimer's disease can be linked actually by actually sleep. So, but we still need to work a lot. But I'm very interested in kind of finding how AD, epilepsy, sleep can be kind of connected. So. Yeah, so with that, I'd like to thank my lab member and actually Dr. Mark Woodruff, Johns Hopkins, who is my postdoc mentor, and technical support, of John Monaco from NIH, and obviously, Elixirgen Science and funding. So yeah, okay. So thank you so much, and happy to take any question.
Thank you, Dr. Tabuchi. I appreciate you. Thank you so much. So for our next guest speaker, and actually just as a reminder, he just mentioned it, please send over your questions. You can do that through the comment, or you can do it through the email at questions at elixirgensci.com. For our next speaker, we have Dr. Cara Gordon. She is from Vala Sciences. She got her PhD in neuroscience at the University of Iowa. She got her postdoc at the University of California, San Diego. And she's currently working at Vala Sciences utilizing their flagship kinetic image cytometer, otherwise known as the IC200 KIC, with high throughput automation and analytics in combination with our iPSC-derived cells to measure single-cell response to drugs and in disease conditions. And she currently leads Vala Sciences' efforts to create iPSC-derived neuron and neuron astrocyte microglia co-cultures to model neurological disorders for drug discovery and neurotoxicity screening. So without further ado, Dr. Cara Gordon.
Thank you so much, Ryan, for allowing me to be on your webinar this morning. Thank you, Dr. Tabuchi, for your interesting talk. I'm excited to tell you all about the work that we're doing at Vala Sciences, modeling epilepsy and neurodegeneration in iPSC-derived neurons using kinetic image cytometry. So at Vala Sciences, we are a CRO that's focused on developing human-induced pluripotent stem cell models that can be used for drug discovery and drug toxicity testing. We routinely use iPSC models for both cardiac and neuro projects. We believe that iPSCs have the advantage of being a human model in a dish, and it allows us to use patient-derived and disease-specific lines. We can set up simple monocultures of neurons alone or build more complex CNS systems with neurons, astrocytes, and microglia by differentiating multiple types of cells from the same iPS line. These differentiated cells will then retain the human and patient gene expression profiles, and they also display electrophysiological properties, calcium signaling pathways, in addition to forming synapses and networks. There are a number of cellular phenotypes of disease and drug toxicity that are amenable to imaging assays, such as mitochondrial defects, neuronal function, aggregated, modified, and mislocalized proteins, and synapse number and neurite integrity, to being a few. And at Vala, we have worked to develop imaging assays to address these phenotypes in disease states such as Parkinson's disease, Alzheimer's disease, for drug-induced neurotoxicity and cognitive decline, and as I'll talk more about today, epilepsy. So to perform these imaging assays, we have developed a small footprint, high throughput imaging instrument that we call the IC200 kinetic image cytometer, which I'll just refer to as KIC throughout this talk. And we have an analysis software platform called Sightseer. And together, the KIC and Sightseer allow for ultra-fast videography and high throughput analytics of single events in single cells. So we can grow simple or complex cultures in 384-well dishes, and then we can perform live kinetic imaging assays or high content assays with the IC200 KIC. And then we can follow this with automated analysis with our Sightseer software so that we can quantify single cell phenotypes due to disease or drug toxicity. Today, I'm going to focus more on our live kinetic assays that we use to investigate neuronal function. So extracellular and intracellular calcium are important for neuronal signaling and function and influence multiple cellular processes. One important role of calcium is that it can enter neurons during an action potential through voltage-gated calcium channels and through neurotransmitted-gated channels when agonists bind their receptors. And neuronal excitability is an important feature of epilepsy, which is a neurological disorder characterized by seizure episodes, recurring seizure episodes, during which there is excessive and synchronous neuronal activity in the brain. Additionally, we find disrupted calcium homeostasis and neuron death through excitotoxicity associated with different neurodegenerative disease. The influx of calcium into neuronal cell bodies can be detected using fluorescent calcium indicators, and we can develop assays to look at the changes that are associated with disease and drug-induced toxicity to the calcium handling, and to assess the rescue of the disease phenotypes with potentially therapeutic compounds. So at Vala, we are routinely running assays with fluorescent calcium indicators in iPSC-derived neuron and cardiomyocyte cultures. And so we're particularly interested in single-cell calcium activity, and I'll tell you why. So this video that I hope that you're seeing here is a video of fluorescent calcium activity in a sparse culture of Elixirgen neurons that were plated in a 384-well plate. The neurons were stained with the calcium indicator ROD4 and imaged at 4 frames per second on the IC200 KIC. This video is being played back at 30 frames per second, so faster than it was acquired. And hopefully under the circles, you can see some flashing cells there. We'll have some better videos later in case you can't see it as well. So the CiteSeer analysis program was used to mask each of the cells in this field of view. And you can see that even though they're sparse, they do fire. They fire asynchronously, and each of the cells has a very different firing behavior. So if we were only to take a well average of what we got here, as happens in some of the imaging uh, platforms that are out there, you that's the average traces at the top. You'd see that we'd lose a lot of information from some of these asynchronous firing cells. And this is why we believe that single cell can give us rich information that could be missing in other platforms. So when we perform our single cell calcium transient analysis, we can extract many different measurements from every transient in each cell in our video. And here is a calcium trace from one Elixirgen excitatory neuron. Because we want to look at neuronal excitability, we're interested in using the calcium as a proxy for action potentials, as the calcium waves are slower and easier to image. So the action potential related calcium waves should have a fast upstroke and then decrease over time. So we have built algorithms in CiteSeer that will identify these fast upstroke calcium transients like you see here. And it can detect the initial spiking portion of the calcium transient, which should correspond to the action potential that is driving the calcium uptick. And so that identified uptick portion is here in orange called the spike mask. On the spike mask, after we have identified where that should be, we can then make transient by transient measurements, such as the spike amplitude or the spike duration, depending on the width of the spike mask. And we can measure the spike area and also count the number of spikes in a cell to look at the number of events in a period of time. And all of these measurements can help us to assess the excitability of neurons. So using our KIC and CiteSeer system, we've done some work to develop an epilepsy model. And this model uses Elixirgen's excitatory neurons. We used the quick neuron excitatory kit and made these neurons in-house with our own iPSCs. And then we cultured them with human primary neurons, and they were cultured together for over 30 days. Then we loaded the cells with Calbright 590 AM, which is another calcium indicator, and captured 5-minute videos at 4 frames per second. And 3 minutes before imaging, we added either DMSO as a vehicle control, or we added 30 micromolar 4-aminopyridine, or 4-AP, which is a compound that will block voltage-sensitive potassium channels. And this should elicit a seizure-like phenotype in the culture. So these are videos of calcium activity from Representative Wells, either for DMSO or the 4AP. And so hopefully you can appreciate here that this is also on a loop, so you might see it kind of look the same and then different things happen. So that the DMSO culture has plenty of asynchronous spontaneous calcium activity. We're not using any sort of trigger to make these fire. But the 4AP, there's a marked increase in the synchronicity of the calcium events that are occurring. And you can hopefully right there, you just saw some flashing. So this synchronicity is a very prominent feature of the culture after we add the 4AP. And you can see that in a video, and we can see it just by looking at the single cell calcium traces. So on the right, we have plotted all of the single cell calcium traces from all the active cells in one well of the DMSO and the 4AP. So you can see that we've obtained hundreds, 230, 242 cells worth of active cell data from one field of view with our kinetic image cytometer. And each of their traces is plotted separately. And so in the 4AP well, you can see groups of neurons that look very similar in transient shape and are clearly firing synchronously, firing together. And of course, it's great to look at it, and it's very interesting to see, but how can we describe, sort of quantify the correlation between the synchronicity in this culture? So in this case, we've performed a cell-by-cell cross-correlation analysis. So every cell is compared to every other cell in the well to obtain a correlation coefficient so it can show how correlated that pair of cells are. So a higher number is a greater cross correlation, and a lower number or 0 is no correlation. So this histogram shows a plot of the correlation coefficients of every pair of cells in these two wells. And you can see that when we add 4AP, we shift the histogram to the right, increasing the cross correlation between cells so we can kind of quantify the synchrony. And we continue to try to develop better methods to look at culture. Cross correlation between neurons and also neural circuits in the well. All right, and now we wanted to look at the changes in transient measurements and culture activity from this experiment. So we performed the analysis of the spike mask as I described earlier. So to look at the changes in the measurement of the transients that we get out of active cells being cells that we were able to identify a transient in, we measure both the amplitude of each transient and the time duration of the spike mask. We can report this both as a mean amplitude and duration by averaging all the spikes from one cell, and then we can also say pick out the largest or longest spike in each cell and report the maximum amplitude and the maximum duration of that one tallest and longest spike. So when we look at this in this DMSO versus 4AP co-culture here, we can see that addition of 4AP increased the maximum spike amplitude, but it didn't actually change the mean spike amplitude. And there does seem to be a trend towards a decrease in the mean spike duration. Now, when we look at measurements of the neuron culture activity, we can look at the fraction of active neurons, which means we're looking at the number of active neurons that had spikes, over the total number of live neuronal nuclei that our masks identified in the culture. And additionally, then we can make measurements from each cell on the total number of spikes, the total area of the spike mask, and the total duration of the spike mask, all over the total time of the data acquisition. This helps us to kind of normalize it versus time, because if you took a 5-minute video versus a 10-minute video, of course, you'd probably see more spikes in the 10-minute video, but now you can compare data acquisition from different time periods. So when we add 4AP to our culture, we see a significant increase in the fraction of active neurons in that culture, as well as an increase in the spikes per second, the area of the spike mask, and the time, the time that the neurons were active during the scan. So here we've been able to quantify both an increase in culture activity and synchronicity after treatment with 4AP, our seizure-inducing compound. So, a goal of ours in epilepsy research would be to develop a robust model using iPSC derived isogenic triculture. So, this would be neurons, astrocytes, and microglia that were all derived from the same iPSC line cultured together. And this would be especially interesting in the study of a genetic epilepsy model, because it would be great to have a better CNS model, a complex CNS model with all cells derived from that same genetic epilepsy donor patient or CRISPR-induced line. So here I'm going to show you an experiment where we have made an isogenic triculture model and treated that model with 4AP also, and phenytoin, an anticonvulsant. Which phenytoin would block voltage-gated sodium channels. So the system that we used were to, again, make quick neuron excitatory neurons from the SEV kit in-house, and then we also co-cultured that with induced astrocytes and microglia that we made in-house. When we treated this system, we used 2 lower concentrations of 4AP than I showed in our last experiment with a 2-hour treatment. And we treated with phenytoin at 15 micromolar and 150 micromolar for 24 hours, and again imaged for 5 minutes. So here I'm going to show you the same spike measurements and neuron excitability metrics that I showed you with the last experiment. And in this situation, we're seeing when we look at the measurement of the spike amplitude and spike duration. With the 4AP, we don't actually see an increase in the spike amplitudes or changes in the spike duration as we saw in our last experiment. But here we are seeing a significant decrease in the maximum spike amplitude and a trend to decrease in the mean spike amplitude with the phenytoin at the highest 150 micromolar concentration. And there may actually be a trend towards an increased mean spike duration. However, it's not consistent enough to gain significance when we compare it to vehicle by ANOVA. And then when we look at our metrics of culture excitability, we see, again, no effect from the 4AP on any of these measurements of culture excitability. And so we think that this is probably due to the fact that we changed both the concentration of 4AP and the time course of addition imaging for this experiment than in the last experiment. Perhaps the effect is only very short, and that we needed to treat it more soon, close to the time of imaging, to see the excitability that we expected. But here again, we do see that the 150 micromolar dose of phenytoin decreased the fraction of active neurons, the spikes per second, the area of the spike mask, and the fraction of active time very much. And so this would be what we expected from the anticonvulsant drug. So as we continue to develop this isogenic model, we need to perform more testing of doses and time points and different drugs so that we can really work out this isogenic triculture model for epilepsy in a robust way. So the last slide that I wanted to show you on neurodegeneration, as it's been a topic of discussion this morning, we are also very interested in building models of disease with genetic links. And so here we have used the Elixirgen excitatory neuron kit to make neurons that we cultured with human primary astrocytes from iPSC lines that were an isogenic CRISPR pair, where we had an ApoE33 donor line, and we've CRISPRed in the ApoE44 mutation that is a genetic susceptibility for Alzheimer's disease. And so here we just wanted to demonstrate the multiple things that we can do with our cultures and our kinetic image cytometer. So first, we've taken these lines and we've performed kinetic image cytometry for 2 minutes at 4 frames per second with our calcium dyes. And you can see that both of these cultures have very nice synchronized but non-induced spontaneous calcium activity. And then we can fix the cultures after we do the kinetic image cytometry, and we fix them and stained for beta tubulin 3 to identify the neurons and GFAP to identify the astrocytes. And if we needed to, we can then overlay these two images so that we could identify transients coming from a certain cell type. And so these are very robust cultures, and we look forward to phenotyping this system further and also other disease-linked systems in the future. So in summary, at Vala, we believe the combination of high-throughput single-cell data and iPSC models will build predictive systems for diseases like epilepsy and neurodegeneration and more. And so if you're interested in knowing more, we'd love it if you would visit our website and contact us. Thank you so much for listening to my talk and for the invitation to speak this morning.
Thank you, Dr. Gordon, for your talk. And that brings us to the Q&A session. So I'll bring back Dr. Tabuchi as well. And if you put your questions in the comments, you can do that, or you can email your questions with the slide number to questions@elixirgensci.com. Doesn't look like we have any so far, but I'll give you guys a minute to post your questions and we'll get those answered. And yeah. Doesn't seem like we have any questions coming in. No worries. I guess we explained the data too well, so all the questions were answered. No worries. So I suppose we can move on. And really, that's the end of the webinar. So I just wanted to thank our speakers for their very interesting dive into their data. It's two really innovative ways that our customers are using our, and I hate the word customers because it makes it sound not as collaborative as it really is. But two really unique ways that these guys are using our cells, and we hope to continue working with them and hopefully work with you all in the future. We will be, Elixirgen Scientific will be at the 27th annual meeting of the American Society of Gene and Cell Therapy right here in Baltimore, Maryland, next week. So we hope to see you there. Come swing by our booth, say hi. Our team will be talking about our newest innovations, technology, and we'll have a bunch of information on the stuff we're working on now. So swing on by, and thank you all for attending. I really appreciate our two speakers, Dr. Gordon and Dr. Tabuchi. Thank you so much. And if you want to learn more about Elixirgen, please visit our website, our LinkedIn page. You can view us on social media or email any questions or ideas that you have to info@elixirgensci.com. And yeah, thank you so much. I appreciate our speakers, and have a good rest of your day and have a good rest of your weekend. Thank you. Bye-bye.
Thank you.
Thank you.